your microphone during the talk. All right. And then the second one is feel free to drop any question related to the topic today. And students, I would strongly encourage you to participate during the Q&A session. Don't get afraid to give any kind of question, because there's no such thing as a stupid question, okay? And another common thing, because we're attending a seminar, so you deserve a point. So to earn the MMA and also the USM CPD points, I think right now we already have share you a link. Please complete the link, click the link and then complete the feedback form in order to get the points. Okay. So now, Dr. Gunn, I guess Dr. Gunn is around.
Yeah, I'm around.
Okay.
I'm just muting myself. Yeah.
Okay, shall we start now? Okay. Assalamualaikum and a very good morning to my fellow colleagues and also students. Our honorable invited speaker, Dr. Gunn Han, a Chief Scientific Officer from GeneSEQ Sdn Bhd. So for those who haven't heard about GeneSEQ Sdn Bhd, it is actually one of the few companies in Malaysia that is offering end-to-end next generation sequencing, the NGS. Okay, the NGS services. All right, so it is my pleasure actually to welcome everyone again to the fourth webinar series talk 2021. But Dr. Gunn, before I introduce yourself to the audience in this room, allow me to also introduce my department to you. Okay, so we in the Department of Pathology, we're actually quite busy for routine diagnostic work for anatomic pathology, but we also do research, which is mostly on cancer-related research. I mean, last year, we were fortunate, we were very lucky to get a research grant actually from university that enabled us to perform 6 cancer research studies that incorporated 2 main analyses, FISH as well as the NGS, which both of that analysis are actually relatively new to us. Okay, so having you with us today, we hope we can actually gain more knowledge about NGS. And to participants, Dr. Gunn is a local, our Malaysian NGS expert with an excellent track record. His PhD is in microbiology, and he has been involved in NGS application for 10 years. And I would say that he also, impressively, he has more than 200 high impact publications in NGS work. Very impressive work there. And previously, he was responsible for setting up the genomic facilities at 2 universities, Monash University Malaysia and Deakin University in Australia. Okay. And he currently joined in the industry about 2 years ago with the goal of giving his expertise to the community. All right. So for today, Dr. Gunn will deliver a topic entitled Using NGS in Research and Clinical Diagnostics. And we hope that Dr. Gunn, you will be sharing with us the key points on the principles of NGS, on how to design the NGS workflow from A to Z, and then also the importance of post-data analysis in NGS. Okay, so without further ado, let's welcome Dr. Gunn for his talk. Dr. Gunn, the floor is yours.
All right, thank you very much, Felicity, for your time and for help introducing me a little bit. So without further ado, I will first share my screen just to make sure that everything is A-OK. Just want to keep it as informal as possible, meaning that if you have any question during the talk, just feel free to ask lah. Apparently there's student as well. I wasn't aware of that, but it's good lah, because that means that we will be able to share information with the younger generation, which is kind of like one of the important goals, I think, in Malaysia, generally speaking, given the landscape of the current landscape of NGS. So in this talk, the focus is obviously going to be more heavily oriented toward clinical diagnostic, but there will be quite a bit of a good introductory slides as well on somewhat how to design, or rather to ensure the success of any given NGS project. I will probably kind of mention a few examples here and there just based on my previous experience. But generally speaking, most of them probably will stem on how I actually operate my lab and how I kind of deal with projects during my time in Malaysia as well as in overseas lah. So let's kind of move on. First thing first, I'm actually a local Kelantanese, so it's good to be somewhat virtually back to my home ground. I still have a house in Kelantan actually, so I haven't been back for a long time. I did my secondary school in the SMJK Zhonghua lah, actually, and then I got a JPA scholarship from the government to go do my undergrad in the United States. Then I came back to Malaysia to continue my PhD in UTM. So that's where I get to be more kind of accustomed to the work culture as well as the research culture in Malaysia, which I feel is very important lah, to kind of set me up to take up the role of empowering future Malaysian researchers in doing NGS as well. Because if you have your training overseas, then it's very hard to understand the key limitation as well as the advantages of research being done in Malaysia. So I actually am quite grateful that I took a very weird path lah. Most people would think that it's a very, very unwise decision, because when you do your undergrad in overseas, usually they would say that might as well just continue everything until the PhD overseas. But I think it's okay lah. Yeah, there's no biggies. In fact, I think I learned quite a bit. So anyway, just a quick overview of my life after PhD is that I actually work in Science Vision Sdn Bhd, which is surprisingly the Illumina sole distributor in Malaysia. So from there, actually, I learned quite a bit on NGS, because my PhD has nothing to do at all with NGS. I do a lot of cloning, a lot of bacteriology work, so microbiology lah, most of the time. But there's no such thing as NGS back then. That was back in 2012-ish, when NGS is just beginning to take its footprint in Malaysia. So as a result, it's like you learn from beginning lah. You start from scratch. There's a lot of somewhat, not to say literature, but you have to read the sequencing manual and interact with the client. You have to act like you know everything lah in front of the client, because my job is actually as a field application specialist, and then become a field application scientist. So you gotta know your thing. You gotta know the technology very well, so that if they have any question about the system, you can actually answer. But the stint with Science Vision didn't last long, because I think I felt that I still have a lot more things to learn, and I can only do that in a less restrictive environment. So I actually then went back to Monash University. Also went back, but went back to academia, not went back to Monash, but I went back to academia to learn and to get more exposure, I would say, to different NGS pertaining to more projects that relates to NGS. And then that's where I learned quite a bit on bioinformatics as well. So if people were to ask me about where I learned all these things, actually it's all based on, nobody taught me. I actually just have to learn it late at night after office hour, and sometimes just Google things up. So a lot of things has been self-taught. So my NGS experience, I would say, is a little bit unconventional lah. But I actually appreciate those times, because when I look back, I realized that if I didn't go through that period, then I wouldn't be where I am today also. So after Monash, my boss actually went back to Australia, so I also followed him back to Australia lah. to kind of have a little bit of more exposure with international, to gain that international profile actually, because I want to test myself on how well I can get when I go overseas, but presenting myself as a postdoc researcher kind of thing lah. That's when I get a lot more exposure in Oxford technology. So Oxford technology is new, but now I think it's starting to gain a lot of foothold, especially because of the COVID situation in Malaysia. I will kind of talk about that as well later on, given that it has quite a bit to do with the clinical diagnostic. It's a whole genome sequencing of viruses. So I'll walk into that later on. And then after that, I decided to come back to Malaysia. Well, honestly, because the contract is ending, I don't feel like renewing it. And secondly, I want to go back home because home is where everything is. But I don't want to go back into academia anymore because, to be honest, I'm more of a free spirit kind of guy. I like to do things my way. But when you're in a university or in any given company that is not set up by yourself, then you're always going to be controlled, or you're going to have limitation in terms of what you can do. So I decided to take like one of the craziest leap in my life, investing in setting up a NGS company in Malaysia. The reason is quite simple. When I look back in Malaysia, I see that the NGS has not been done right at all in Malaysia. It leads to a lot of problems in terms of the sustainability, in terms of the lack of expertise, despite the fact that NGS has been around for so long, because of the way things has been handled from the NGS point of view. I think in Malaysia, we need to improve upon that. So I decided to come back and hopefully can share and also work together with local researchers actually to build up our profile once again. So my experience, as you can see in my Google Scholar, I mean, it's not like 200 very high impact paper, not it's just, we have publication, but it's not like each and every one of them are in like Q1, no. So just want to correct that a little bit. But we work a lot on NGS based work. And then as you can see here, even the top 4 most cited paper has to do with NGS. It's mostly, surprisingly, it's 16S. So you can see studying the microbial community is quite a heavily researched topic nowadays. And I think it also relates to a lot of clinical application as well. With that, I will now slowly go into the technology and whatnot. Let's see if I can move, overshoot a little bit. So when I was in Monash, we actually did something. We actually sequenced one of the first fish genome. We also try to expose students, undergrad students, not even the postgrad, but these are all undergrad students to sequencing technology, because I believe that's where we need to put our focus on, because the undergrad student will then bring forth the passion in NGS in their future undertaking, hopefully in their PhD as well. What I realized though is that most of the time you learn things in academia, but if you want to apply it, you have to go to industry. So it actually partially explains why I decided to take this leap also at the end of the day. So because of the COVID, there's not much we can do. Probably that is why you never see me out in the wild also, because you can't go out. So we have been trying to do this kind of a virtual talk, as well as just do things, more R&D stuff in GeneSEQ Sdn Bhd. So, I mean, the COVID thing has been a great lesson also in terms of how you can demonstrate the power of genome sequencing done right. So that actually got some attention from the media, which I will probably share with you later on as well. What we realized though in Malaysia is that a lot of the company, although they say they're offering NGS, they're actually not doing NGS. They are just middlemen, meaning that they take your sample, they send it out. Even the report they don't do themselves. They ask the other person to do the report for you. Then they just send the report back, and then whatever difference between the cost from the provider and their, like the market price that they sell to you, is their profit margin. It's simple as that. The problem now is that the value is not there. You're not getting a very I would say comprehensive service because of this. Because if you have any question about the report, for example, it's very hard to get back to them because their job is just a middleman, meaning that they don't really they are not heavily involved in the sample actually. So even if you have any question requiring consultation, they are not the go-to person. And then if you want to go ask the person who did the analysis, it's not in Malaysia, this person is somewhere else outside overseas. So this creates a lot of an issue. One of them obviously being the language barrier as well. So that's why I realized we really need to do things very locally. And then I realized somehow there's no such person yet in Malaysia. Then I decided to be that person to take this leap. I mean, it's so far to me it's a very rewarding experience because that allows me to get to know many people in Malaysia who is doing fantastic research, and one of them are you yourself in USMKK. So I'm very grateful for the invitation. So just to kind of bring forth to what GeneSEQ generally has been doing, we are no middlemen. You pass your sample to us, we actually talk to you, talk about the science with you as well. I think that's important because that actually allows proper NGS experiment to be designed. Then it allows the costing and everything to be done right also. There's no overestimate, there's no underestimate. The number of sample sequence should be just about right to fulfill the statistical requirement. Because everything is done, well, most of the things are done in-house data analysis, sample QC, as well as sequencing. That means that if somebody would like to pay a visit to our lab, you can always come and have a look at how we're doing our stuff. You can have a look at how we are storing the sample also, because everything is done in-house. We didn't just take the sample and take it somewhere else to have it analyzed. Obviously, we also do a little bit of outsourcing, but the outsourcing part is only when we need a lot of sequencing data. Then we have to outsource because why? It's cheaper. But the majority of the work after outsourcing, we get the raw data back. We also do the data analysis in-house so that when people ask about how the data was analyzed, we can actually answer to them. And importantly, that I realized is that what we want is also local talents. Meaning that you don't want masala already inside your paper, and then that's what I'm actually aiming for. We should be able to be independent, even though it's gonna be like a big, high-profile genome paper. We should not need external people to do it. We should be able to stand alone, providing that we know what is the value of our research. So as a result, I really value, or I totally encourage local collaboration. Most of our projects with local researcher has thus far turned into more of a collaboration rather than service, because I think that's better. It's a win-win thing, meaning that when you collaborate, you actually share and exchange knowledge rather than it's just a one-way thing whereby you give us money, we give you the report. I don't think it's very healthy also, that relationship. Collaboration means that after the sequencing work has been done, project has been completed, the relationship still remains. Meaning that we still talk from time to time about current project, future project, as well as student training, for example. So I think all these things is required from a local NGS company. Gone are the day that, service are just service already, my opinion. Because if those things still remain, Malaysia NGS is not going to grow properly or healthily. That will be my take. So as you can see here, we have done COVID genome. We have done even the Empura, like a very expensive fish in Malaysia. Actually, to be honest, there's more to come. It's just it hasn't been published, so I cannot put it out here because we also have to respect the P&C of our collaborator. So there are more than that, definitely. But what I'm trying to say is that people are actually open to collaborating with GeneSEQ Sdn Bhd. And I wish that would be the unique point that makes us very different from any other NGS company in Malaysia. So now I go to the actual the basic things. It's not gonna be super duper detailed because I think that will require a very project specific understanding. So what I'm doing here is something very, very, I would say very generic, but I think it's better to start off as generic as possible before going to overly detailed. So for any given NGS project, you need 4 main things. If you don't have these 4 things optimally like selected, then an NGS project either will not even succeed or it will ends up eating more than the required amount of time as well as money. So I just gonna quickly go through these 4 elements in this particular presentation. So first thing first, before I even go to that, I just want to share with you a few remarks that I heard from Malaysians dealing with a sequencing service provider. People always think that NGS is slow and expensive. I think it's partially not true. It actually depends on how the thing was done. And it has to be done properly. Then it's actually worth the money as well as worth the effort and time. Service provider like to send a lot of data and flood people with a lot of graphics and picture in the report and expect us to understand what it meant. I don't think it works that way actually. What this means is that the Bioinformatics Foundation, at least from us Malaysian researcher, we actually need to achieve a certain level of bioinformatics literacy first before we can actually interpret the result properly. So what this means is that the bioinformatics component also is important. But I'm not saying that you have to be like absolutely an expert in doing all the command line and terminal. No. But the basic needs to be there first, like understanding what a certain key parameter means and what's its implication, so on and so forth. That needs to be there. So that's why the bioinformatics elements has been included in the guideline as well. Again, is regarding the analysis. Sometimes when you become more proficient already with the analysis and then you're asking for something extra, people tends to want more money from you. So I don't think that's the right thing. Why? Because that indicates that the consultation or the discussion hasn't been done beforehand. Expectation has not been managed properly. So if that's the case, that means that the project either has not been fully disclosed or the supplier or the service provider don't really understand the goal of the project. So it all comes back to the sample as well as the project objective. This actually pertains to sample as well as the sequencing machine of choice. Like when you have a sample that keep getting rejected for not passing QC, actually I would say there are a few reasons. Number one is that people in general are overly conservative with how they do their QC, meaning that they require a certain number, like a certain value to be achieved before the sample can actually proceed for NGS. And obviously we have people saying that, rubbish in, rubbish out, those kind of thing. I personally feel that sometimes you can find value in rubbish as well, because in my experience, I also work with like sample that other people has thought of as rubbish, but I managed to extract information out of it. So a rubbish, like you see all these paper cups and the plastic, if you go put it to the recycling plant, you still can get money out of it. I mean, it's the same thing with the sequencing result, but the standard has been set, sadly saying that, oh, if your sample is like, oh, the DNA no good, looks very degraded, then nobody want to sequence it for you. That usually is like a rule that somehow has been set by the service provider, and it's also sometimes set by the people commissioning your sequencing system. But by and large, I don't think that's true, because there are certain applications that actually can tolerate, for example, degraded DNA, as long as it's not overly degraded. So I mean, it's still rubbish, but it's not very bad rubbish kind of thing. So that requires a lot of experience as well as confidence in your sample. So at the end of the day, the sample types obviously matters, but there are certain samples that memang cannot get good quality DNA. For example, sample that has been stored for a long time, then if the storage condition is okay, then maybe you will get less degradation. But give it some time, then at some point in time, the degradation will accumulate. So with that in mind, before even starting the project, knowing the sample condition is good, because that allows you to know what kind of strategy that can be chosen to deal with what kind of sample. And for those working on statistic, obviously the sample numbers matters. But if you have too many samples, then your sequencing cost will go up also, because each of the sample will require X amount of data. And you have too many samples, then it's too much data required, and then there's going to be a lot of money involved. So sometimes we can think of something along the line of pooling the sample and then doing sequencing as well. So this is something that we go with the name of pool-seq. Or sometimes you can just focus on a specific part of gene, because that's the gene of interest, and then perform NGS or amplicon-based sequencing on it. So that will work too. My take is that starting a project, don't be overly ambitious. Oh, I want to do 100, I want to do 200 sample. I wouldn't go with that first. It's always a good idea to start small. Don't over invest in the project first until you get a better feel of it. Don't over promise actually. You want to start small, especially if you haven't done a NGS project yet. You want to start small and use that as your stepping stone to just kind of experience the entire journey of NGS. And once you have enough confidence, that's when you go a little bit bigger. Then keep going up and upwards. So that's, I would say, is the strategy lah. So sample wise, again, storage, number of sample, how you want to start it, that all actually matters. We probably have been staring at the rubbish for too long, so I'm going to change the picture. This one is nicer. Okay, so if the sample is available, then you are able to extract either usable. I wouldn't say good quality, I would just use the word usable. Because as long as something is usable, lower quality also never mind. As long as it can make a sequencing library, then to me, that's all that matters. Who cares if the thing is very intact? If it cannot be used to generate a sequencing library, no use also ma. So we shouldn't judge a DNA by its gel image lah, or its its nanodrop or whatever reading. Not overly, because to me, if your DNA is okay, usable, then why not go all the way with the library preparation? So the definition of library preparation is pretty much either converting the DNA that you extracted or the RNA that you extracted into something that can be kind of run or inserted into the sequencing machine. And that usually involves slight modification of the DNA fragment. Like just put example for Illumina, say you want to do whole genome sequencing. That means you have to break the DNA into very, very small pieces, because the Illumina is working on short read sequencing. So you have to break it into small pieces, and you have to do ligation to attach a specific sequencing machine specific DNA adapter, which will then be recognized and bound when it goes into the sequencing machine. So this actually involves very fundamental molecular biology work. For example, end repair, for example, ligation. It even has like a PCR step involved during the library preparation step. So it's not rocket science per se, so don't be intimidated by the NGS workflow. At the end of the day, it's nothing special. It's just typical fundamental molecular biology workflow. All right, so just want to put some focus on the point form, right? The first thing is obviously the money. What kit is cheap? What kit is not cheap? What I realized is that, or at least in Malaysia, my observation is that somehow, unfortunately, if you buy the branded one, which in this case is actually the Illumina, the cost tends to be slightly higher than the other brand. So what I'm trying to say is that even though you have like an Illumina machine, right, it doesn't mean that you must use all Illumina product. Illumina product tends to be more expensive because they're premium. And then obviously it's more expensive because unlike other brand, they have their technical people locally to provide the consultation. But also this, I think, is another thing that needs to be think about because of the location. We are in Kelantan, but your tech people is in KL, meaning that it's quite hard for them to provide 24/7 face-to-face support at your site. So it's something to kind of put into perspective as well lah. If you happen to have like a sequencing system in USMKK, something to think about. I think that will come back and bite actually. The ease of use meaning that certain kit is longer, they have more steps. Certain kits don't have that many steps. So it depends on, well, number one is how technically competent the student or yourself will be in terms of doing the library preparation also. So you see, if the step is easily understand, it's important that you understand the step as well. Not just follow the protocol. You gotta understand, okay, what is this step doing? Why is it doing it? And if you understand that, at least you understand the basic of it. Don't just follow bulat what the manual has to say. Just gotta understand it first, and then interpret it yourself. Think whether or not, okay, is it something that we can actually scale up and do without much human error at the end of the day? How much samples are we comfortable working on any given day to avoid like sample mismatch or using the wrong barcode, those kind of things. This all depends on how detailed is the manual. Sometimes overly detailed manual is good, but it also introduces some rooms for error. The definition of availability, right, means that how fast can you get the kit? If a kit will take, many months to get it, that means that the supplier is not prioritizing the kit, or the kit is actually in high demand. But usually it's the other way around lah. The supplier don't really prioritize that kit. Most of the time, like certain kit, because it has to be imported from outside. So what that means is that the business people or the company selling the kit will want to strategize. They will always try to find a good time and batch it, meaning that they will consolidate many orders from many clients and then one shot order it. So you have to be, if a project is very emergency or important, you might want to make sure that you're choosing the kit that is more flexible, meaning that the supplier can provide it faster, or if you already have a stash of it, meaning that you have some of the backup kit, reaction kit already with you for any given time, so that if there's any emergency project requiring sequencing, you don't have to wait months and months just for the reagent to be available to you. So that's, I think it's very important who you choose to work with as well. Just for the record, GeneSEQ, we don't sell any like NGS reagent yet. I don't think we'll be doing that. So what I'm telling you is just based on my experience dealing with the local suppliers around Malaysia. But so far, we seems to have something in the work lah. Suitability is pretty much like choosing the right kit for the right sample and for the right project. You got to make sure that, oh, if I want to do whole genome sequencing, this is the kit that I want to use because it can generate good quality data at the end of the day. Third party has a lot to do with availability. Third party meaning that you don't rely on the direct product that was sold by the Illumina company. There's many enzymes that has the same function, and then you can actually use that as well. So what I'm trying to say is that you should not, or at least for myself, when I was still in Monash, I was not penalized for using third-party reagent to make the Illumina library. Why? Because even the Illumina people say actually there's no problem with doing that. In fact, the entire world is doing it sometimes. So meaning that not all company, university, or sequencing provider is using just Illumina brand to do their library preparation. As long as the final sequencing library contains the necessary component, it can be sequenced on a given machine one. So this is just something to be mindful of, meaning this is saying that you can actually choose from quite a good number of different company to process your DNA or your RNA into sequenceable library lah. QC is very important because you really got to make sure that the library preparation is okay. So the job of QC is to ensure that, okay, after you settle all the library prep, the final product is actually suitable for sequencing. So this involves, to put it simply, quantification. So let's say if I input 100 nanogram of DNA for my library preparation, but if something somewhere went wrong along the line and I only ended up with like 0.01 nanogram of DNA, that is a problem. I wouldn't even want to put it into the sequencer already, because that gives me an indication that maybe somewhere along the line, the library preparation may have failed, leading to wrongly constructed DNA or insufficient DNA to proceed. So that's not a good sign. Actually, you want to have sufficient final DNA library or amount that kind of looks correct. Too little is no good. Too much is still acceptable, but that means that you over amplify also the library. So you want to strike that balance, and that value, right, is usually will be embedded in the sequencing library preparation manual as well. Another QC that people can do is they will run it on the gel, or nowadays got the bioanalyzer, the tape station. We personally don't invest in that because it's very expensive. The consumable for bioanalyzer is very expensive, at least to me, as well as the tape station. What they do is pretty much a very mini gel and very sensitive gel, because it's run at a very small lane. Imagine running gel electrophoresis in a lane that's less than 1 millimeter or 1 centimeter like that. So it's going to be very intense and concentrated. So that's what the QC will do, because if you can see smearing that at the right size and the concentration is good, because the smearing band is very intense, and it's a good sign. So QC kind of work that way. It ensures that you got something to sequence. That's the important bit of library QC. Then come the interesting bit, the sequencing system. I just want to take this opportunity to say, currently, right, in Malaysia, I felt very sad when I come back, because what I see is a lot of these things in Malaysia, and like every corner, a lot of the sequencing machine, for some odd reason, was not actively used after they purchase it. It almost seems like people have the money to buy the system, but they are either not investing enough to build up the staff to manage the sequencing machine, or that there's not many sample coming in. It almost seems like there's some missing part or missing link in getting the thing or getting the lab and NGS lab to be up and operational. So what happened is that because it's not well managed, not well maintained, then towards the reviewing time, company, or rather the university people upper management maybe see that it's not generating like it's not generating return of interest lah, so to speak. So shut down the lab. So it's pretty sad because it's a big investment. Certain system can cost up to millions and millions of ringgit. Some obviously it's not that expensive lah. Some could be just 500K or 200K, but I think that's still money going away. And the sad thing is that every day you'll be seeing that system just shut, like it's not turned on, just sitting there gathering dust. So I really hope that we can kind of reduce that at least in the near future. What has been done has done already lah, meaning that those systems that's decommissioned already is unlikely that they're gonna revive again. So we just have to kind of work with what we have. That's the way it is, lor. Just kind of putting some perspective into why this is happening as well. Based on our experience, certain equipment NGS has annual maintenance fee. So before starting a project or something like that, you might want to check whether the thing is still under warranty or not. So most of the time, they provide free warranty in the first year, but after that you have to settle it yourself lah. And usually it's not that cheap, and like it can cost up to 50K or even 10K, 20K, at least 30K lah, I would say 30K a year, just to kind of make sure that everything is in order. But again, just want to highlight that if you are far away from the company, what can they do to help you? I feel like most of the time you'll be on your own rather than having like a shoulder to cry on kind of thing. If there's something wrong with the sequencing, right, it's not like you're gonna get instantaneous help. They have to take a place to actually go to the place to check it out to check what is the problem. Obviously, you can share screen and do all this remote troubleshooting, but at the end of the day, the lab operator in the university or in the lab is still gonna be the one who is doing a bulk of the troubleshooting. So that is something to consider also in any given project, because you really got to make sure you got everything covered. Okay, if you want to start this run, at least make sure that there's nothing wrong with the machine. And then if, touch wood, something went wrong, you know who to talk to, meaning that you know who to have a chat with. Can you get replacement? Most of the time, when there's a run fail, right, what we want as a customer is a replacement reagent. You don't need to replace us for the time that we have to spend or wasted to troubleshoot for you, but at least please give us a replacement so that we can restart our project. See? But sadly, what I found out is that if they found out that the problem is with you, meaning that the library got some problem with the QC and things like that, you don't get any replacement. So they only replace it when they really confirm that it's a problem with the reagent, not so much your library or whatever, or if it's a problem with the system. So lesson learned is that you got to do your QC properly, and then if there's a fail run, right, you have to make sure that it is a reagent or a system problem. If it's a system problem, you better make sure that your system warranty is still there, because if it's not there, then you're not going to get any replacement. There is this kind of hidden warranty on the reagent, but not on the machine. So let's say if the run failed because the machine shut down halfway through, right, and then you do not have a warranty in place, sorry, that's totally your loss already. So that brings another thing lah. You really got to make sure that you have backup power supply and all these things, and ensure that everybody is keeping an eye on the sequencing when it's running. So there's no such thing as just start the run, walk away, and expecting come back tomorrow everything is sunshine and rainbow. Yeah, there's like this Murphy's Law thing lah, somehow with this Illumina system, in my opinion. Something somewhere, something might just go wrong. You just got to keep an eye on it to make sure that everything is in place. And then just remember lah, like us researcher or user, we want to save the amount of reagent, but it doesn't work the other way because they want you to buy more reagent. So you can see there's a conflict, like the conflict of objective lah at the end of the day. So be very careful with what people recommend. Like for us, we always get reminded, oh, do you want to buy reagent? You're running out already, right? Do you still want to buy more reagent kind of thing? But they never ask, oh, how's the machine? Are they doing okay or not? They just say, you want to buy a reagent or not? So these are the things you have to deal with, lor. To me, it's a bit pushy marketing, but it is what it is, lah. So they have to, find revenue as well, so cannot blame them. But this is something to kind of think about. There's this difference in the perspective when it comes to sequencing. So you have to be very careful with who you're working with as well, in terms of the NGS run project and things like that. But I just want to kind of highlight that we don't just use the Illumina anymore nowadays. We are starting to put quite a bit of interest. So this has to do with, how do I put it? This has to do with breaking away from over-reliance on a single system. The more you rely on just one single system, you can think of your neck as being tied by that company already. You don't have that system, you cannot do sequencing, right? Meaning that if you only have one single system, they can do whatever they want. They can exploit you. They say, oh, we don't want to support you. Then you can't do anything about it. Then at the end of the day, you got no sequencing project. So you really want to avoid having just one investment in a single sequencing technology. Just be open to what kind of technology to use. Nowadays, we have Illumina, we have PacBio, we have Oxford Nanopore. I personally put Oxford Nanopore because why? It's low capital, meaning that you don't need to pay 100,000 ringgit just to buy this small device. The market price, I cannot lie to you anyway, because they display it publicly in their domain. To buy a starter pack, I don't sell it. I'm just sharing you the procedure lah. Is the starter pack costs around 1,000 USD, and with that you get the device as I shown here. You don't get the laptop lah, but you get the device. You get the flow cell, pretty much quite a number of library preparation kit reaction as well, that allows you to actually start a small mini sequencing project. And it costs around 1,000 USD base price. But when you import to Malaysia, there's this 10% import tax as well lah. So just so you know the procedure of kind of acquiring a nanopore. They don't have a local agent. I don't think they are playing that game also, because when you have a local agent, the price go up even more because you have to take care of the local agent. So they do it based on a more research community approach, meaning that everybody work together, support each other. I think that's the way I want to see NGS actually grow in Malaysia as well. This is us kind of like spending some time. This is like what? Maybe in 2020. Last time the COVID got, last time we get some very hardcore lockdown because of the first ever cluster. Then they are looking into faster way of sequencing the COVID. So we actually work together with I think this is at I think. Yeah, so we work with the group to help them to do the nanopore sequencing. We also have the Illumina system, but as you can see, they also invested in the nanopore system just because I have quite some experience running the device. And then what I did was I actually paid a visit to their lab with the laptop that I actually custom built in my company one for doing a nanopore sequencing. Go there, set everything up, and on the same day, we are actually sequencing the COVID-19 genome already. In fact, what's shown on the screen is actually the sequencing coverage of each of the COVID-19 genome, the genomic segment of the COVID-19 genome. We did not sequence the entire transcriptome. We actually amplified and enriched the virus genome, and then we sequence it. So it's very different from what I suspect the current practice is right now in Peninsular Malaysia. But in any case, it's something to consider as well. What I'm trying to say is that there are more than one way to achieve a certain NGS objective nowadays. And then you'll be surprised by how much more advantage or benefits that you can get from being able to run multiple small sequencing projects. Because the minion, right, the cost of the flow cell is not to say that expensive. And then they have a smaller version of it, which is very suitable for a small project. So the upfront cost is less than 1,000 ringgit Malaysia, and then you can do a few bacteria genome, whole genome sequencing. That's the kind of a cost that we're looking at. And it's obviously it's long read and real-time data. I'll talk more about the real-time later on. This is just showing you that there's more than one way to do NGS already nowadays. Another thing, this is the part on real-time lah. So when we say real-time, when I put the library into the device, less than 10 minutes, I can start to get data that I can use straight away, you know. I can use that straight away to see whether or not the sequence belong to what virus, which region of the COVID-19, for example lah. But the problem with this big, chunky machine is that you have to wait, and it's not a short wait lah. Usually it's nearly 19 to 24 hours before you can get any kind of data set. If you terminate the run halfway for Illumina, then bye-bye lah. Everything is gone. You totally burn your data already. Pretty much wasted the entire cartridge. The nice thing about Nanopore is that you can stop anytime you want, you can pause anytime you want, and then you can resume the run, and you can still use the data. So it gives a lot of flexibility. And that's probably why people like to bring Nanopore to all the jungle lah, to the desert lah, to the forest, all those kind of places to do their sequencing, because it's very flexible. And I think Nanopore, because it's real time, meaning that you can get data to be analyzed very fast, it's great lah, because that means that it's a very nice tool to use in the clinical setting when you have emergency case. In fact, if I'm not mistaken, I heard that people have used this Nanopore to sequence like those tumor tissue or tissue biopsy when the surgery is still ongoing, you know, just to make sure that they have eliminated a good majority of the so-called tumor tissue. If I'm not mistaken, I read about that before. So to me, that's very, very amazing. You cannot do that with Illumina sequencing because it's not real-time data set. You have to wait. Okay, I did not expect everything to come out in one shot. I want to slowly go through it one by one, actually. So never mind. So now we go into the bioinformatics and data analysis. All these pictures actually relates to each other one, actually. So first thing first, when we're doing bioinformatics or data analysis for NGS data set, right, you can actually say goodbye to Windows lah. Usually people do it in the Linux operating system. You can actually install the Ubuntu nowadays on your laptop, on your Windows laptop. So I think that's a good thing, because that means you don't need to buy a brand new computer, just install the Linux operating system, meaning that even a student, undergrad, as long as they have a okay laptop, which nowadays is quite okay lah, the spec, like you got 8 GB of RAM and some sort of Intel i3, i5, you can actually do some, I would say, not so intensive bioinformatic analysis already, meaning that I can probably do bacterial genome or viral genome assembly on a laptop, because the software has come a long way, meaning that people have improved on the algorithm to reduce the amount of memory usage. So that's great, because that means that you can actually do it on a smaller system. Ubuntu is usually our choice for the operating system. And if you were to go to the Windows software store or whatever store, right? If you type the word Ubuntu, it actually will show up. So we actually, at least on my laptop, I'm using Ubuntu 18.04 lah. But we also kind of at GeneSEQ lah, we provide some sort of help to set things up and then give some very starting basic training to get more involved in the NGS data set, so that you always know that, as you can see in the middle picture, there is a personal trainer. So we prefer to train personally. What that means is that we don't really like the idea of, wow, today one workshop got 10, 20 participants, everybody learn bioinformatics. That to me is nonsense one. Nonsense, because everybody have their own project. Everybody have their own learning space. There's no such thing as, oh, I go to a bioinformatics workshop that I know how to analyze my data set. No such thing. because bioinformatics is not a one-day thing. I mean, Rome is not built in one day. Bioinformatics lagi lah teruk. Means that you have to actually slowly work at your own pace. And then the most important thing is while you're training or while you're embarking this journey, that somebody next to you, so-called a personal trainer. So that's the kind of a training that I think is important. To not ever give the data to somebody who don't understand the biology to analyze, because that will end up hurting the entire project. You as a researcher, not asking you to know everything, but should know a little bit of the basic. And then the more harder things to execute, the harder software, for example, then maybe you can pass to the other guy to do it. But you should be able to interpret the result after that. So some basic in bioinformatics is very important. And you can see the cat that has the octopus tail there. That thing is called GitHub. You might have heard about it when our health minister talk about it. That's where all the KKM COVID data now has been hosted. So this GitHub database is a very nice repository for free and open source and peer review bioinformatics too. So what am I saying here is that do not buy those licensed software, because number one, they are not peer review. We don't even know how they work. So the result come out, sometimes looks good, you happy lor. Sometimes don't look good, you also don't know whether it doesn't look good. Well, we still happy lah, because it's very fast and easy to use. Licensed software tends to make it very, very user friendly, to the point that you just have to click here, click there, and then it works. It might sound enticing and very interesting at the start, but the problem is that you're not learning, you're just clicking, you see. You're not learning in the sense that you don't understand what happened at the background, but this is very important to understand also. You may say, oh, because last time nobody kind of guide you, now we are here lah. So if you think that that would be the way forward, or you want to go down this path, now GeneSEQ is always here. And I realized that this is actually missing in Malaysia, and I think that's one of the bigger reasons of why we are not advancing also in NGS. A majority of people who so-called have this bioinformatics degree or in PhD, right? They are actually from the time when protein modeling is very hot topic. But protein modeling and NGS is two different things. You can use NGS to predict the protein to do modeling, but you cannot do it the other way around. You cannot use protein modeling to do NGS. So as a result, there's a gap. Just because a person called themselves bioinformatics, you gotta know their root. If their root is during the docking stage, molecular modeling, blah, 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 then it's not really NGS. So if you ask them to do NGS also, it will take some time before they actually understand the data and whatnot. They might be good programmer, but they may not understand what biologists want to achieve with their NGS dataset. So that's that. And because there's a lot, I think the bioinformatics for NGS now has matured quite a bit. So I'm actually seeing quite a lot of good quality software, somewhat very user-friendly also, in the GitHub repository. So I think it's very, very promising. The time is good. The timing cannot be any better to start learning bioinformatics. But don't learn bioinformatics for the sake of learning it. Learn it because you want to use it to apply to your project. Because there are many different tools, and then you are not able to understand them all. You got to know what tools to use to achieve the project objective. Once you have that, you consider yourself having a pipeline already, like a way forward to generate from raw sequencing data all the way to the report or whatever visualization that you want. That's the ultimate objective actually. I show a big computer here for a reason lah. Two things. One is storage. When you have NGS data, you have to always think about backing up your data, meaning that you might want to invest in a few, I wouldn't say very high quality hard drive. It's more like you want to have multiple copies of your data. Backing up, meaning that it's not about having very high-end hard drive and things like that. It's more about having multiple copies of your file in different hard drive. I'm not saying that in the same hard drive, you create a new folder called backup and then you put everything there. No, the backup folder has to be in a totally different hard drive. It's a totally different hard disk lah. Then that's called backup. Because if somehow something happened to the first file, you actually have a backup somewhere else. Ultimately, you want to put them all in the cloud lah, whereby you will have like NCBI, SRA, you're gonna have the accession number. That would be the safest lah, because once you have that, in fact, sometimes it's the requirement for publication to make the data publicly available so that it can be peer reviewed as well. So obviously, while you're analyzing it, you're still unsure how to publish it. You can obviously have it in your hard drive, but just make a few copies of it. For example, you can have the actual working hard drive to do the data analysis, but then you can make a copy in a separate hard drive, or you can make a copy in like a portable hard drive. Because my experience that I've seen hard drive failing, meaning that people make backup in that portable hard drive, they assume everything will be okay. They don't have a copy in their original computer anymore. Then comes the day want to do the upload. Wow, the file was corrupted. Then it kind of messed up the entire project already. Raw data, always remember, the raw sequencing data is a very, very important NGS currency. If you don't keep a copy of that, you will not be able to reproduce your work anymore. Raw data is like raw material. No raw material, no product. You cannot regenerate the product from, you cannot regenerate raw material, raw sequencing data from the final product. No, it doesn't work that way. Last but not least, on the lower right, Google. Means that you got problem, you Google first. That's what I'm trying to say. I learned a lot actually from Googling, because there's a lot of what we do is we will notice an error when we're running something, and then we'll copy and paste the error in Google and then see whether people have reported that before. Like most of the time, actually 80 to 90% of the time, the error has been encountered by somebody, and then a solution has been proposed already. So what I'm trying to say is that it's not like very scary to do bioinformatics. It's just you need to be resourceful, and then now you have people guiding you. I think things will be much more streamlined as well. So don't rely 100% on those data analysis companies, so-called, to just do the analysis. Importantly, also is sharing and understanding the sequencing result lah. I think that's the end part of the 4 elements. All right. So I just want to bring this into the application base now. We are doing clinical diagnostic, at least in USMKK, this department lah. So to me, I'm trying to think of what can be done. Cancer is interesting, but also we have bacteria infection, outbreak, and I think all this kind of thing also plays a role. In fact, like I personally have this experience whereby my brother-in-law passed away because of sepsis. So it kind of make me feel that, wow, if I know, or because that time I was in Australia, I can't do much also. But I just kind of think about it, like if I can apply, like for example, the Nanopore real-time sequencing to help to identify the root cause, could I have changed the cause, how things progress or not? Because like he was suffering and the doctor was a bit busy doing other things, then they just suspect only after he passed away, they do post-mortem, then they kind of say, oh, it's due to the sepsis or like secondary, so some sort of infection lah. But it seems to be bacteria related. It was not captured early, but he also has some issue with his white blood cell platelet count. So I'm not sure whether that also contribute it lah. But I just sometimes think to myself, if this thing can be detected earlier using NGS, would that have changed everything? Because he's quite young, actually, he's only like 30 something, less than 30 if I'm not mistaken. Sorry about that. So, and then he has a son, so I felt very, very bad about it. So yeah, I mean, sometimes I feel that if I were to apply NGS properly, maybe things could have been much different lah. So anyway, back to this is that whole genome sequencing is good, because if you can sequence the genome, you can do many things. One of it, I think last time we heard about this food poisoning thing in Terengganu. I don't think I heard much about the genome information. about the strain that's been isolated. I'm not even sure if they identified the causative strain or not. But so that's that. What I'm trying to say is that if we can do whole genome sequencing, you can get a lot of information. And for example, you can get their species ID to a very, very high accurate level because you're not using just the 16S. You actually can identify them based on their genomic characterization, the genomic profile. So it's high accuracy genome-based species ID, which is good because knowing what you're dealing with allows you to know how you want to combat the outbreak or the disease, what antibiotic to use, for example, to kind of deal with certain species. Maybe certain antibiotic work better, this kind of thing. Another thing is that because you have the whole genome, complete genome, right? Meaning that all the potential genomic potential, all the possible antimicrobial resistant gene can be mined from the assembled genome, which is good lah, because the last thing you want is to use an antibiotic that this guy already has resistance to. You're wasting it, and then actually overdosing a patient with antibiotic is not a good thing lah. Do you know what happened? It's like my brother-in-law, they found that, oh, this guy maybe got, they suspect lah, got this bacteria infection. Then they dose him, not to say overdose, but they dose him with antimicrobial antibiotic. Then the next day he passed away. So it's very, very sad actually. So I'm not sure whether, if knowing the correct antibiotic resistant gene, will that have helped, or could that have changed things? Because most of the time it's like a broad spectrum antibiotic and then hoping that, you know, things will go away rather than a more targeted approach. And rapid MLST pretty much means that you are able to kind of know it's a sequence type. Sequence type meaning that it's like a combination of mutation that separate strains from the same species. Meaning you can have a certain E. coli from different sequence type, and then this sequence type maybe is more pathogenic, or they are more they can adapt more to certain environment, those kind of thing. Because when you have the whole genome, right, it pretty much all the information is actually on the silver plate for you already. You can learn a lot about that bacteria before taking action. The question has always been, how fast can you generate the genome after you have it in the pure culture, for example, or if it's still in like a broth culture, how fast can you get that information so that you can take it to the next step? Serotyping is quite much like MLST. It's like additional information from the genome you can get, but it kind of relates to the how you can better identify them, because certain bacteria strain people use serotype rather than MLST to differentiate them. One good example is the group B Streptococcus, group B strep. It is much more relevant in terms of serotype. People actually differentiate them based on their serotyping. It's like based on their capsular, what the capsular polysaccharide, those kind of structure lah. You can actually predict that by sequencing the entire genome and then understanding the gene that relates or associated with the serotype displayed by the bacterial strain as well. Last thing is phylogeny. You have the genome, you actually construct the phylogenetic to understand the evolutionary relationship. I mean, it's pretty much the same as how people are tracking the COVID, because when you accumulate a mutation, that means that you're slightly different. So by comparing the number of mutations against the original strain, you can see how far it has evolved and how closely related it is to the original outbreak. If genome is too big, we can also do targeted amplicon sequencing. The targeted amplicon sequencing, the way it works is that rather than sequencing the entire thing, we only sequence the region that is of interest. A very good example, which has been done very commonly or routinely now in like Malaysia or every part of the world, is the 16S amplicon sequencing. Because 16S is a gene that is present in all different species of bacteria, and they are actually different among different species. So what you can do is that you can sequence this small fragment of gene, and then you can use that to infer the microbial community, which is something good when you're investigating a certain infection or want to just monitor the gut health, gut microbiota, those kind of things. It can be done very quickly. So that's bacteria. But don't forget, You can also do something similar in human. You know this gene probably have some mutation, and that could lead to cancer, for example. What you can do is that you can design primer and amplify out the region and then go for sequencing. Because we know cancer is like a mixture, you have a mixture of normal cell and cancer cell. It would be nice to know the percentage of the SNP that is associated with cancer relative to the percentage of SNP that is considered the normal, that's considered to originate from the normal cell, to just have an idea of the heterogeneity of actually the cancer cell population. So this can be done using amplicon sequencing as well. It's not something that you can do with Sanger, because Sanger is not something we use to identify like rare mutation. Rare mutation, you need high throughput, and then you want to sequence the same thing over and over again because they originate from different cells. So if you have one cell that is a little bit different from the rest because you're doing it at such a high throughput, then you can find that mutation of interest, and then you can say, oh, we found this SNP in only 1% or 0.1% of the population, the cells population, not the human, but the cells population, the tissue. So this can be done, and it's much cheaper, and the analysis is also much simpler because it's just a matter of aligning the sequence read into the reference gene and then just count how many times you see this SNP and how many times you see the other SNP. So it's a very, very much easier, I would say. Next. Oops. Another thing we can use amplicon sequencing, this is much more relevant, is to do the COVID genome sequencing. What you may not know is that if, like in the very, very beginning, I don't know whether it's still being practiced right now. What I read from the news is that the COVID virus was isolated from us human, and then it has been then enriched by putting it into a tissue culture. I think the Vero cell or something, Vero cell, to make more copies of the virus. Then after that, they sequence everything, including the human transcriptome, including the host genetic information. They do it together with the thing. Essentially, they just extract whatever they can from the tissue culture that has been infected with COVID. And then they hope that in the entire data set, maybe 0.01% of the sequencing data belongs to the COVID genome. It's a little bit of a brute force approach to me. And then amplicon approach came along. What it does is that it has a set of primers that enrich, or rather that amplify only the COVID genome. Because the moment you have a genome sequence, right, it's a matter of time before you can actually design overlapping primer to actually fish out the entire genome. So the COVID genome is 30 kb, which is quite long actually. So, but right now, if you look at the global database, the majority of the sequencing, or rather the COVID genome, has been obtained using this approach rather than the conventional Vero cell, and then followed by brute force sequencing. So I mentioned about this because I know when they do this amplicon-based sequencing, is usually done on the Nanopore device, not so much on the Illumina, surprisingly. But I also still kind of wonder to myself what is being done currently within the government. We have been shut off, just so you know, like GeneSEQ and like industry generally. We don't know what exactly is happening in terms of the genome sequencing. We can only say things from the outside, what we felt is actually relevant. So we don't know what's going on right now. There seems to be an increase in the amount of genome from COVID recently, but it's not in Peninsula. I think it's more from the Sarawak side. So it's quite interesting. It'll be curious to know what technology that they're using, what method that they're using to achieve that. Because to have suddenly increase, that means that usually technology has changed. It's not that suddenly the lab management has improved dramatically. It doesn't work that way. Usually it's a technological breakthrough that allows people to do it at a higher throughput. So that remains to be investigated. Okay, before I go on to the next slide, I actually saw a comment from Muhammad Suhilmi on how actually the genome database by NGS can assist in the development of personalized medicine? Okay, this is a good question actually. Genome database, essentially what happened, you need to have some sort of a population genomic information first. So personalized medicine is like we have specific drug or specific, I would say, diet catered based on the person genomic information. In Malaysia, this is still very far-fetched, sadly speaking, but it doesn't harm to be the first one to give it a go. Okay, genome, if you sequence the entire genome, very expensive, and then the data is overwhelming. But if we know in our Malaysian population, this is another problem also, because Malaysia, we are more genetically more heterozygous. We have way more difference in the race profile compared to the European or the American population. So what happened is that the mutation that you found in this individual, in this particular population, may not be the same in other population. So that actually imposed a problem. And secondly, is because there's not much genomic database for our own Malaysian to allow us to do a much more precise genome-based design for personalized medicine. However, that being said, if we design like a panel of gene that is known to be associated with personalized medicine, for example, maybe a resistance to a certain drug is always associated with this gene, for example, then you can actually design the primer to target that, and then you can have another primer to target another gene that kind of interact with a different kind of drug. Because I heard about warfarin resistant, or like there's some SNP that is associated with this how an individual reacts to warfarin, the blood clot or something agent. Sorry, I forgot about that. But I know about that, and like it helps a lot to understand what is the mutation that the individual have before administering that medicine. So rather than saying it's a genome database, it becomes more of a targeted gene database. But for that, you need to know the patient or the individual background, how they react with this drug first, and then you have to know their genetic background. Then from there, you can design, oh, this panel is good. It will help to identify this particular mutation that actually can be used for personalized medicine in the future. And personalized medicine is one thing, and then also diet law, because what you can do is that you can check on you can actually check on a certain gut microbiota to assess how healthy is the individual, and then adjust the diet based on their gut microbiota to strike that balance. So certain bacteria actually can be used as a biomarker for their health indicator as well. So that's something to kind of look into as well. Okay, we got 2 questions from Hilda, which is the range of library preparation kit that's recommended to use, as well as what's the recommended library concentration for QC. So my take is that it's quite technical. The range of library preparation kit, actually, you have Illumina, you have Kappa, you have quite a big range of the brand. My take is that it all depends on what you want to do, use it first. Like if you want to do whole genome sequencing, then maybe a safe approach I might recommend is a bit pricey, but it requires the least amount of equipment would be like those enzymatic fragmentation kit. So they have the the Nextera Flex or DNA library prep kit now from Illumina, or the New England Biolab. They call it the FS library preparation kit. So these two doesn't require a lot of equipment investment, and then it can be used to do whole genome sequencing, shotgun sequencing library. That's for whole genome. And then if it's for amplicon, you actually don't even need those kind of kit. You just need a PCR polymerase and the correct primer. And the primer, we are more than happy to share with you because this is open knowledge one. So you don't even need the kit to do this kind of thing, because the inclusion of the sequencing adapter, everything is already in the primer design, which make lives much easier to work with. the recommended library concentration actually most of the time is very low. It's not high at all. It's like in the range of maybe just 1 nanogram plus minus. In fact, to put it into perspective, like our Illumina system only need 100 picomolar. Pico is like what, 1 to the 10 minus 12 molar of DNA. Very, very little only. But if you want to just do QC, I'm not sure which part of the which part that you're asking, but like for sequencing, you don't need a lot of the library. QC wise, I mean, what QC will do is it will tell you the concentration, but when you're doing QC, even though you don't need a lot for sequencing, the QC actually reflects on how well is the sequencing library. So my personal opinion is that if you're doing like just a quick concentration measurement of your final DNA library product, at least aim to have at least 1 nanogram per microliter. More is better. Anything less than 1 nanogram, it might still work, but just kind of have to be a little bit more careful because it indicates that the library preparation is not very efficient. Because a good library preparation means that you're going to have a lot more, I would say, a lot more concentration than more than 1 nanogram per microliter. Okay, we got another question. More the important. Oh, I mean, this is like the most important thing to do. To have a genome database in Malaysia. The problem now is that everybody is doing their own thing. Nobody actually come together to come up with a dedicated genomic database. What would be nice is that anybody who partake in this particular genome database should, whereby we need to know their background, ethnicity, all this kind of thing, health condition, and then we perform certain sequencing. It can be a genome, but it will be very expensive, but it could be a set of genes to kind of just kickstart the entire project. And then once you have that, you got the statistic. Certain mutation will have a certain correlation with a disease outcome or diagnostic outcome. Then with that, things will be easier. It has to start from somewhere. And I cannot emphasize how important it is. We are totally lacking behind. We always talk about want to do the first human, not to say the first, but like some human genome in Malaysia. That was like how many years ago? And I haven't seen anything happening. Now you can see everybody is sequencing human genome like nobody business, but we haven't done to that level just yet. Because I think it's not, this genome sequencing for human database, this kind of thing, I think it should be a continuous project. It shouldn't be like a grant that lasts only for 3 to 5 years kind of thing. It needs to be continuous. Why we want this database is because when you have the database, it's like a book, you know exactly, now that you have the book, you can actually correlate certain mutation to certain disease, certain diagnostic outcome. But without the book, you're going blind. You just say, oh, maybe this is why this guy has this problem. That's about it. But if you have a database, it's a totally different ball game. The problem now is just that the database developed for the Eastern, the Europe, like it has been heavily developed from that, doesn't really apply very well to our current population. It's just the way things are in terms of our genetic pool is very, very different. Okay, so I think that's that. I'm going to proceed with my slide. So just want to kind of talk more a little bit about working with more than one type of bacteria. Is that in any given infection and whatnot, usually you can have more than one types of bacteria, especially in your gut. You're gonna have a lot of different kind of bacteria, and if that's the case, the striking the balance is important. I like to talk about gut because my focus is a little bit on microbe, but I understand that it could be a little bit different. So I'll just make it as fast as possible so that it's not so suffering for everybody. So balancing the microbe is important, but how do you know your microbe is balanced? You have to sequence it. So as a result, the old way is that you do qPCR to quantify, very limited, because you're not getting the entire picture. You're just getting a rough quantification, semi-quantification of the composition. So with NGS, it's a totally different thing because you're actually sequencing doing like applicon sequencing. You can actually identify whatever that's in there. You're not detecting a specific thing anymore. If you have how many % of certain pathogen, the NGS dataset will give you that information right away. You don't need to have multiple PCR assay just to achieve the objective. So this is actually me sequencing my own gut microbiota. I will not share with you how I sample it, but I will share with you my sequencing data lah. We did that internally just to prove that using the Nanopore, not the Illumina, we can get the same outcome, or I would say a more improved outcome lah. I think the top one belongs to me, B2 and B1, and the bottom one belongs to my daughter, who is 1 year old only. But you can see there's a very big difference in the abundance of the Bifidobacterium bifidum. Daughter has more, I have less. Apparently, this bacteria, it will go down when you start to age lah. But maybe I should eat more Bifidobacterium bifidum punya probiotic, which actually is available to actually improve the to kind of gain back their to get the balance back. Who knows? But just showing that once you have this information, you can start to take action. It's the same thing with having a genome database. If you know more information, then you can do things better. Now everything is in the dark, you're just guessing. Oh, maybe my stomach not feeling well, but I'm not too sure what's the problem. But if you were to sequence, for example, the microbial community, you can actually start to see a difference. And then it's not just sequencing one thing, you have to do comparison. That's why a database is important, because it's not going to just have one thing, but it's going to have from different population, different individuals. Then you can do the comparison to find out what is actually significantly different. The nice thing about this particular data set is that rather than sequencing the V3, V4 region, a very small fragment only of the 16S, we actually use the Nanopore long read technology to sequence the entire 1.5 kb 16S RNA, which gives us much better resolution. So to bring things back into perspective, it's nice because that means all this while last time maybe certain human gene can be 1,000 base pair or even 5,000 base pair. It cannot be sequenced well on the Illumina because it needs to be prepped. There's a lot of work problem. And then you cannot do haplotyping because everything has been broken into small pieces. But if you can sequence the entire intact long amplicon, then you can do proper haplotyping as well. So you can actually identify what mutation is linked. Are they linked together or not? Certain SNPs, is it always together with this particular SNP? Are they linked? This kind of information you can actually gauge from Nanopore sequencing. And obviously, I don't need to kind of say that, it's so much easier to run it on the Nanopore because it's such a small device and it's easy to interpret as well. That's that. So actually, this is actually kind of like bringing things back lah. And then I'll open up for question. We want to have good sample in order to have good NGS project, but the definition of good is just something that's usable. Don't like super cannot use degraded stuff. As long as it works, then the project can jalan one. Don't worry too much. Don't tie yourself to one technology. Be open to other sequencing technology. Even if it's not available yet with you, there's always people with you can collaborate to make it work. Really over-relying on one technology, at least in my experience, spells trouble. It kind of takes away my ability to bargain as well. Bioinformatics, it will keep coming up. You cannot stay in one place with bioinformatics. You gotta keep exposing yourself to new software and how they work, and can they actually make a significant change in your current NGS project or not. Be on the lookout from time to time on this kind of bioinformatic database or bioinformatic software, for example. Investing in a machine. This can be a curse or can be a blessing depending on how it's being utilized and how it's being managed. Because the last thing you want is to see a machine that has been decommissioned, and to reactivate it back, you need to pay a lot of money. So something to think about, because sometimes you just want to complete a small project. Then it doesn't mean that you need to invest in buying the entire machine just to finish that project. Sometimes it might be better to reallocate that resources to the actual project itself. I know that, okay, it's nice to have the machine because you can physically see it, but sometimes it's not just about that machine. You need to have the right team to make sure that the whole thing is oiled up properly, all the gear, everything, all the component, people receiving the sample, prepping the sample, everything is done right. It's not as simple as having a machine. Let's just put it that way. But obviously, if this thing is up and running and then you're churning out data day in, day out, then perhaps it's a blessing. But always bear in mind about the cost of the sequencing. Bigger machine costs more, but it's cheaper to sequence on the big machine. Small machine like ours, we only have the Illumina iSeq 100 is like by far the smallest. But we get very little data from the system, and then every time before we want to start a round, we have to pay around 4,000 ringgit Malaysia just to get that little amount of data. But for the same amount of money that I'm paying, I can get much more data if I sequence it on the big machine, big Illumina machine, much more, like 100 times more data, possibly. So that's why I say outsource is not a bad thing, but you got to make sure who you're outsourcing and who you're working with. And secondly, sometimes disease doesn't wait, virus doesn't wait. They know no rule, they don't care. You cannot say, oh, stop replicating. Doesn't work that way. So how to sequence to identify the problem or what system you use actually matters. So sometimes certain things require a lot of data and then has no like critical waiting time. Sure, you can use Illumina, but if it's a matter of life and death, or like it's more serious, it's good to know the resistant profile, for example. It might be better to choose another alternative technology that can give you the result quicker. To batch or not to batch pretty much means that sometimes you're not just sequencing sample from your own project, because it could be very expensive. So what people like to do is that they share the reagent so that you split the cost across different collaborator. So that's something you can think about as well. It has its pros and cons, but generally it saves money, but it may introduce conflict as well, because some people may not like to share their sequencing run, even though it can lead to a less cost to their project. Last but not least is the short read versus the long read. Don't forget, Illumina is only like 150 maybe to 250 base pair, the sequencing read. But with Nanopore, you can go up until 10,000, 20,000, no problem. So your amplicon can be as long as they want, as long as your PCR can generate the amplicon. Then you just need to pop it into the Nanopore sequencer, and then off you go. I mean, to sequence the entire 16S ribosomal RNA for bacteria, can't do that with Illumina. You have to use the long read sequencing technology to do it, because it's too long already. So, I realized, at least in GeneSEQ, by keeping us flexible, it actually brings a lot of benefit and flexibility, and allow us to kind of work more closely with many local researchers. So that's that. Okay, so last but not least, talking about collaboration and project discussion, always open for that. You have any question about NGS and things like that, we're always happy to discuss. This is my phone number. You can just double check. Hilda also got my number, so can verify that it's real. Not like some random guy will pick up the WhatsApp, reply the message. So, and then, my email and my company, the company punya email. So that's that. Okay, I will now have a look at more questions. We have another question from Hilda about the FFPE tissue sample. Yes, you are right in the fact that FFPE sample tends to be more degraded. So in fact, FFPE sample is not something that even a sequencing company want to deal with. So most of the time, you have to kind of make sure that you do the proper preparation. So you just have to go in with the mindset that if I have like 100 FFPE sample, not all of them will pass through the library QC. So that would be my take. Importantly is you have to figure out a good way to extract the FFPE to make sure that the DNA can be recovered. It will be super degraded, that's for sure. And then we usually have this method to deal with it by repairing the DNA damages. So there's this like FFPE kit from NEB that helps to polish up the so-called chemically modified DNA fragment. And then after that, you proceed to the normal library preparation. The nice thing about FFPE is that they're already degraded, so you don't need to kind of fragment them anymore. You can just prepare them and then use it directly for sequencing. It tends to be a little bit more complicated, I would say, for the RNA lab, but still there's always kit available to work on this lah. So we're happy to kind of discuss, I guess, further in details what is actually the project details and the outcome that you guys are looking for. But generally, anything FFPE can be considered as so-called the problematic sample lah. So that's that. So, another question that I actually missed out is can Nanopore allow sample pooling? Yes, no problem. I think I sequenced, there's one time we sequenced nearly 24 samples in one shot. You're not limited by like the number of samples, but how much index barcode that's available. Thus far, Nanopore provide around 96 indexes. So meaning that you can do 96 samples in one go, which is like a full 96 well played already lah. No issue with that lah.
All right.
So, okay. Another question is that, what's the problem when you don't have the bioinformatics analyst expert in the research team? Actually, it's not a problem because what happens is that you have the data generated, but you don't know how to kind of move forward with it. However, I also want to stress that bioinformatics analyst expert, this term,
Mm-hmm.
who are they actually? You see, what is their background? Do they actually do the NGS data, or they just day in, day out do programming? This is very important to differentiate. You want somebody who understands the biology, but not somebody who just do programming day in, day out. Just because they have the name of bioinformatics doesn't mean that they are doing NGS lah. Just like I mentioned in my slide just now. I mean, if I know how to help, then obviously I will help lah. I just gotta make sure that I understand the project. So far, so good. Project that we have with academia, so far the deliverable, everybody seems to be happy, because what I normally do is I try to simplify the terms and things like that to them. It's like giving a data demonstration as well as making them understand what this data set means. So far, my best result is I managed to teach a 50-year-old plus professor about the assembly lah, and he understands. So I think that's really good, meaning that the explanation is simple enough for them to understand, because never ever rely 100% on the bioinformatics analyst. I think that's the number one taboo in NGS. You cannot put 100% trust in them. You gotta somewhat know also a little bit on your data set. So that's that. Yeah, I mean, bioinformatics course, in fact, yesterday we did a very quick one-to-one training on somebody from UCSI. So we are continually looking for this opportunity, but we also have a rule saying that we only do very serious one-on-one training when the person has data set, because it doesn't make sense to train you everything and then after that you go there, you go on and kind of explore the data yourself. We prefer to work directly with the data that you have in hand, so that you achieve your objective, and then you also learn and being trained along the line. So we offer that kind of training lah. So thus far, we also do it remotely lah. Understand also now traveling is still somewhat restricted, but the remote training, as again, is like one-on-one, one-on-two maximum, because we do not want to dilute the value of the training.
Okay, Dr. Gunn, thank you. Okay, I guess, I mean, for myself, I have quite a lot of take-home messages today, like, you know, selecting the true sample, relying on the third party for library prep, as well as space, learning for bioinformatics, right? But actually, I'm also interested to know more on the, what do we call this, the sequencer maintenance, you know? Because I must say that I agree with what Dr. Wali mentioned in the chat box. It's something related to consider on how long the system can be supported by the company, and then be aware that actually the new model always come out very quick, you know, as time goes by, and as a result that the older system cannot be supported anymore. And in fact, to the extent that the company cannot even supply the parts of that sequencer, right? Yeah. But my question is that, based on your experience, I mean, I mean, I'm sure that you also have experience on this kind of matter. I mean, what are the best solution for us to overcome this kind of limitation?
My take would be that always go with the mindset that no system will last forever. That's number one.
Okay.
Okay. Meaning that you give a system like 5 years time. That's the business term, 5 years.
Yes,
After
time.
5 years, consider the system gone, kind of thing. So if you're not willing to have that mindset, or the upper management cannot accept that mindset for NGS, then don't even buy the NGS machine. That's the ultimate truth lah. Have to be very, very frank. In fact, like I buy the iSeq 100 now already 3 years. So 2 more years I need to stand by lah. Buy a trash can to throw inside already lah.
Sorry, Shafrina.
Yep.
Uh,
The what? Cannot mute one.
Shafrina, boleh ke you mute yourself? Shafrina.
I think okay already. Okay. Anyway, back to the question. So, yeah, if a system can last 5 years, consistently maintained, then it's great. Certain system I realized from the Illumina one lah. Certain system like the MySeq seems to last longer than everybody else. The HiSeq, like the one in MGI already, I can guarantee already gone case already, meaning that decommission already lah. I don't foresee them turning on the system to run because the reagent also already not in demand, meaning that they are probably not manufacturing it for any longer time. So this is an important thing to understand lah. So that's why my take would be that be very careful when you want to invest in this kind of a heavy NGS machine. And that's why I say lah, I kind of prefer to work with smaller system whereby there's less risk lah, and it's more fun also because I don't need to be so stressed out when I'm starting a run on, for example, on the Nanopore kind of system lah. So yeah, it won't last long. So your colleague is very right to point that out. And then in fact, it's like a marketing scheme lah, because the thing cannot last for 5 years. They want you to buy a new one so that they can earn more money ma. So that's just business at the end of the day.
3 to 4 years, we can actually propose to have a new machine then.
Something like that lah.
Something like
So that's seems to be the situation lah actually. And but then again, come to think about it, it might be better to also not like tie yourself to a certain sequencing machine brand, for example, because now I feel very comfortable using the Nanopore because it's just give so much flexibility in our sequencing in our sequencing pipeline currently lah. And because we don't need to wait for many samples to come in, it actually allows sequencing to start very quickly as well. But when you have a sequencing machine, it's very important to ensure that everybody is 100% on board to use it. But what I realized is that the machine type also affect who will be using it. Because if you invest in a not so expensive machine, what that means is that the data that it can generate very little. So not everybody will want to use it because not enough data cannot proceed with the project. And then if they want to use the big system, it's too expensive to buy. So eventually it might be better to do the outsourcing bit, but have more, I would say, more investment in understanding the data that come out of it lah. So that's why even in my company also, I was like, I just need a machine to do QC lah, and sometimes do some emergency sequencing. If I have a big project, I will still send it outside. And then I just ask them lah, you just give me the raw data lah, don't need to do any reporting, blah, blah, blah. Because I know what to do already when I have the raw data set. So perhaps it might be good to put more investment or time and effort into somewhat building up a foundation for, I wouldn't use the word bioinformatics, but I would use the word computational biology, meaning that every biologist should have a little bit of an experience in computational side of things. Not asking you to write software, but at least you should know what software to use, how to know whether a data is no good, rather than relying on somebody to do it lah. The foundation needs to be there. Because I realized that seems to be the most important bit. sequencing can be done by anybody, as long as they have the system and blah blah blah. That's not an issue. But once you have the data set back, how are you gonna deal with it? That becomes the ultimate bottleneck. And then we seem to have this tendency to somewhat outsource the data analysis, but that actually is not a good idea because the people doing the analysis for you don't know the biology. So it creates a big mismatch between the expectation and the deliverable. So yeah, that will be my take. Not so much on system investment because, eventually it's very sad actually to see all this system gathering dust in the lab. It pains my eye actually because I know in Klang Valley I've seen too many of it. Every lab, like not to say every lab, but those lab that has the machine, we know that it's not being run 24/7 because the lecturer or researcher also said that these people, they don't respond to email, they very slow, things like that. So this becomes a problem. So like if I have given a choice why I want to choose somebody who is not 100% on board to do the sequencing work for me, I'd rather have somebody who can, I pass the sample and then get the result back. So that's that lah.
That's something like that. All right.
Yeah.
Cool. Okay, any more questions?
Yes, thank you very much for the presentation. Can you hear me?
Yes,
Yes, you made mention that actually bigger machines gives you a kind of more data, and then the smaller ones, though they are cheaper, but they don't give much data. But it's like in between, you are trying to provide a middle ground service in which you are in between the major machines that are too expensive and the smaller ones that are cheaper but doesn't provide much data, if I can understand your presentation.
Yep.
Okay. Like most of us know that NGS is very costly, and of course the maintenance. These are very key important things for anyone that is trying to set up a database for NGS. Just like others has asked, how is the cost of continuous maintenance of this? Because it's very critical to be sure of what you are going into at a point, maybe after 1 year when your company has gone and probably one cannot be able to sustain it, then you are going back to zero level.
Right.
Yeah, thank you.
Okay. Yep. That's a good question. So my take is that maintaining the system, at least just like a yearly warranty, it could go up to at least 30 to 50K ringgit in Malaysia. You're just paying them just to make sure that the system is under service warranty, meaning that if you need any help, as long as you're still under warranty, then they'll provide you with the so-called support. So this company has been around for a long time. I don't think they'll be gone suddenly. So I'm not entirely worried about that because we also have their Illumina system. So this is here to stay. It's just that you just got to make sure that if you guys are doing your annual budget, you have to justify why you want 50 to 30K just to maintain the system. So the upper management, at least when I was in Monash, I have to fork up at least I think around 60K to maintain the MySeq. Now it could be a little bit more than that. So it has always been this bureaucracy whereby you have to say, oh, we do so many samples, we bring in so much money, so please give us 60K so that we can just pay the company to so-called maintain the system. So in a university setting, depending on your upper management as well, this is the kind of money that you need to fork out. But that's just paying them. We haven't even discussed about like the staff who should be running the show in the lab. It is not something that you just bring the sample and start the run. It's not as simple as that because first thing first, the people needs to be quite well trained, and some people are still very hesitant to run that big system because they know if something went wrong, the consequence is very dire because it could be that the entire run is like totally gone, that kind of thing. So there's a lot of things to consider when doing this sort of investment. That will be my take. money-wise, it's definitely going to be a money-burning machine. Don't expect to make like revenue out of it, but at least hopefully it should be enough to keep the system up and running. But so far, I haven't seen a very successful story just yet from my side, at least from our claim value side. So I'm quite pessimistic about that, to be very frank.
Yeah, thank you. There's also another question that was asked. Probably you do not clarify much on it, on the issue of formalin fixed paraffin embedded blocks. That's from Pathology. Has a lot of archives of formalin fixed paraffin embedded blocks. Can we really use the nucleic acid that is being extracted from the formalin fixed paraffin embedded blocks for NGS? And how long? Because some of this will be kept for years, maybe 10 years, 20 years. How good is the DNA that is going to be extracted from this formalin fixed paraffin embedded blocks for use in NGS?
I mean, as long as they are, first thing first, you got to make sure that you got some DNA out of it. So I think that's number one criteria. And secondly is how much you get out of it. The more you can recover, the better. So we have seen situation, or rather there has been a lot of publication on the use of FFPE sample for NGS. So it's not like a totally no kind of thing. It's just that it will not be as smooth as a normal type of tissue because of the fact that it has a lot more chemical modification associated with it. So I myself, we haven't had much work. In fact, we have one researcher asking us to do it, but like we kind of just politely reject that because we don't want to disappoint them as well in terms of like making sure that the project is a success. Rather, we kind of work with them to ensure that at least they can get over the extraction side of things first before we even worry about the library preparation. Something can be stored for a long time and still work. Sometimes certain things, depending on how you store it also, it actually cause problem and then it cannot be used. So that's why I said in FFPE, there's no fixed rule. Okay, it's more like if it works, it works. It can just go all the way for library prep and sequencing. A little bit of a luck dependent, at least in my opinion, because there's just so many factors that could affect the success of the library preparation. So the bottom line is it can be done. It's just that don't expect it to be super like 100% success rate kind of thing.
All right, thank you.
Yep.
All right. I think there's one question in the chat box. Dr. Gunn, can I read it for you?
Sure. I think it's about the BGI-seq one, is it?
Opinion about BGI-seq sequencing system.
Right. BGI-seq is actually a China-based company. I think they probably have their support, like their distributor in Malaysia. It's much like the Illumina sequencing. The mechanism is slightly different. My take would be that I haven't used the data per se, but I know that the sequencing, how do I put it, is actually on par. But the thing is that BGI is a very new technology and it's from China. So my concern would be how well is the company representing the BGI-seq in Malaysia in terms of their technical expertise. Because right now, I think they're just looking for people to experiment, to use as their training ground, so to speak. They want you to buy the machine and then use that opportunity to kind of learn from it as well. But given the geography, actually, it could spell a bit of a disaster lah, because after they set it up, they are in KL, but you guys are in Kelantan. So if there's any problem, then things will go haywire quite quickly. That'll be my honest opinion on the BGI-seq. I have no doubt that the data actually looks okay, but to make sure that you generate the data in a setting whereby language barrier could be an issue because it's a China company, not an English-speaking company, that actually will be something that I would actually be quite concerned about lah. In fact, they have actually spoken with us as well, but I politely reject them because number one, I just want to make sure that everything that we're doing actually is done correctly first lah, rather than putting a lot of risk in a sequencing technology that mimics that of the Illumina.
All right.
All right. I have another question before you go. Yes. How do we validate the NGS data?
That's a very broad question, actually.
Yeah.
I mean, there's a few things you can look at. You can look at
Yes.
the sequencing output, how much data that you're getting, the quality. Usually, the quality is not too bad.
Okay.
Also, you can see whether the FFPE sequencing read are actually aligning to the correct region of the human genome or not, or it exhibit certain kind of alignment bias. Then it's something that obviously you have to be worried about. And then you got to make sure that the reads are actually aligning to your SNP of interest so that you can actually use that information to derive, to kind of plan your next move as well. So yeah, I mean, it's just a typical thing. Usually without knowing exactly the objective of the project, it's very hard to give like an absolute valuation of the quality of the sequencing, because sometimes you can have a very lousy sequencing result, but because the sequencing actually managed to identify what you're looking for, it's not so bad after all, because you still achieve your objective. So what I'm trying to say is that you might have a bunch of very short reads, low quality and whatnot, but if like 1% of the read actually map to the region of interest, then is it still rubbish? Yeah, is it still rubbish, or actually, oh, I can move on with my project already? So that will be my comment. It is not like, oh, you have a lot of data. I mean, having a lot of data is great because it increases the likelihood of finding the SNP of interest and things like that. But it also comes with a cost, right? So my take would be to do a very quick test first before going all in with any given project.
Yeah, another question from Dr. Wahyudin. Can we do validation using Sanger sequencing as well? Can we do
I
can
mean,
we
if the
validate?
gene depends on the gene line. If FFPE, then it's a little bit problematic. But obviously, if your target amplicon size is very small, then maybe you can sequence it on the Sanger without much of an issue lah. And then it certainly depends on the project scope also, because like you can't validate a library using Sanger sequencing. NGS library cannot be validated using sequencing, but potentially like an amplicon kind of amplicon based for human, those kind of stuff, maybe you can do it on Sanger, because there's not much mixture of the DNA base. If you're talking about doing Sanger validation on those metagenome 16S, that cannot also, because Sanger sequencing is all just about the pure sequence. That's that. And then the gaps in the Malaysian genome database, well, let's just put it this way. I don't even know whether a Malaysian genome database exists or not. Currently in Malaysia, there's not much marketing, or I don't come across like a strong publication of like a dedicated Malaysian genome database right now. So it might not even exist just yet, or hasn't been set up properly. I'm happy to look at the link if it's available, and then I can give my valuation on that. But to me, a good genome database should have like around in the realm of hundreds or even thousands high quality whole genome sequencing at least.
That's right.
Yeah.
Okay, any more question? Got like 3 minutes to 12. Question?
It's pretty good. I mean, we covered the entire 2 hours. I did not expect that actually. So yeah, it's great actually to see that it's more interactive than I thought.
It is. Perhaps please, just a reminder, please fill in the attendance link and also the feedback form. Okay. Is there any question?
Yes, I don't know, apart from what Walidun asked, validating using Sanger sequencing, can we validate also using qPCR?
To validate whether the library is in good amount, you can use qPCR. To see the content of the amplicon, obviously, it's not something that the qPCR can do lah. So qPCR and quantification actually goes hand in hand. So it's just more tedious and more costly to run a qPCR compared to just a quick quantification of the concentration. Obviously, I don't deny that qPCR will be more accurate as well lah. So it's I would say it's a choice lah. Do you prefer to use qPCR or not, kind of thing. It's good to have, but at least in our lab we don't really do it. Too expensive to buy a qPCR machine. Oh, another question: do we include a control sample follow the disease target in our test that we know? Yeah. You definitely. It'll be good to have a control sample, like those without disease or things like that, as a start, so that you have something to kind of compare against. It's either that, or you have to go with the assumption that this gene is wild type. It's always this particular sequence, but it's always nice, right? Even you're doing research, to have like a proper actual real control, rather than nothing at all. So if obviously the course allow, then include some control. If the course does not allow, then just go with the disease and then compare it with the general SNP database, human SNP database, for example. The global one lah, not the Malaysian specific one lah.
So I guess no more questions so far. Wally, sorry, I just realized that actually it's not a question, it's just a statement. Dr. Wally, I'm so sorry. I wrongly read your statement. It's just a statement, not a question, okay? Okay, so I guess no more question, yeah?
Yeah, I think so.
Just one question.
Again,
Okay, thanks so much, Dr. Gunn, and thanks, Dr. Siti, for arranging this event. Thank you so much. I want to ask, what is the best way to use NGS to discover new mutations in a specific cancer?
In a specific what?
Specific cancer, specific type of cancer.
Okay.
Mutations there.
Okay. So my take would be that if you don't have absolutely no idea what is the gene that you need to target, then just go for a all-in whole genome shotgun sequencing, provided you have the funds. So what you do is you have a normal cell, you do whole genome sequencing from the same individual, and then you sequence the cancer cell as well, and then you identify all the mutation that's possible between these two. And from there, you have a list of SNPs that is only present in the cancer but absent in the normal tissue cell. So that will be number one. And then once you know what gene has the mutation, then you can design the primer to make it more, to kind of do a more targeted approach. So this is on the assumption that you have absolutely zero idea what or which mutation on which gene. Because if you know which gene, or you have a suspicion of what gene could be causing the cancer, then you can actually do a quick amplicon sequencing, which will be much cheaper compared to doing a whole genome sequencing. So whole genome sequencing, think of it like finding a needle in a haystack. It's expensive, but at least you'll find a needle. Amplicon sequencing is like doing a gambling. You kind of have an idea that is there, then you try to scoop it up, but there's a possibility that there may not be any mutation in that gene. So it's all based on your background knowledge and your suspicion and your experience as well as a researcher, and also literature's review, obviously.
Thank you so much.
Yep, no worries.
Okay, any more questions? I guess.
I think
no more questions. Okay.
Yeah.
Right. Thank you, Dr. Gunn. It was really glad, have a talk from a Kelantanese boy. Today, I mean, so-called Awe Kelantan. Okay, but very informative talk. I kind of like your analogy pictures actually from garbage, toolkit, white elephant, Spider-Man, Sheepman, and again with your and also with your last slide of the conclusion. Okay. So it's bringing to a very clear explanation regarding to NGS, what to do before, during, and after the analysis. So I guess everyone here also get a full idea about things related to the NGS. Okay. So on behalf of the committee members, okay, and our co-organizer, GeneSEQ Sdn Bhd, We thank everyone for being here today. Okay, we hope you enjoy this session and we wish you all a blessed day. But don't forget to join next week webinar. Okay, it's something related to implementing continuous community engagement in COVID-19 pandemic. So we will post
Oh wow,
the
it's a
email.
nice one.
All right. Okay,
That's a nice
thank
one.
you, Dr. Gunn. Okay, thank you, everyone. So might end the session right now.
All right. Right. Thank you. For attending lah. I look forward to future collaboration and whatnot lah. Okay.
Oh,
All right.
don't forget one more thing. Don't forget that
Okay.
the session has been recorded, and the recording of this webinar will be available on the department Facebook page. Okay.
All right. Yep. Sounds good.
Pathology PPSP.
Sounds good. Yep.
Okay. Thank you again.
Okay. All right. That should be it. All right. Thank you very much. All right.